Well, thanks, Hud. And my goodness, how far we've come in such a short amount of time. I think it was 4 or 5 years ago that we first had a meeting of the CUG and NGLY1 communities, and it was much smaller than this. I can tell you that. So it's wonderful to see the progress that we've made as a community. So what I want to do today is really give you kind of an overview of the history of NGLY1 and also give you a sense of where we're headed in terms of its future when it comes to things like therapeutics. Now, for those of you who have seen me speak previously, you know that I usually have to give a standard disclaimer because I spent much of the past 2 years actually working for the White House. And that standard disclaimer is that this is a personal talk and this may not reflect the views of the President or the administration. Except that I no longer have to give this disclaimer. And the reason for that is that I quit about 6 weeks ago. So, and people sometimes ask me, well, what was it like being more or less the last scientist left at the White House? And all I could really say on the record was it was interesting. And that's still all I'm going to say for now. So with that in mind, there's really 3 things I want to tell you about today. I want to give you a sense of how far we've come in terms of understanding NGLY1 deficiency and what progress we've made towards treatment thus far. How far do we have to go if our goal really is understanding treatment and cure of this disease? And how are we going to get there? What is it that we have to do to help these children? So how far have we come? Well, first I have to say, obviously, I'm presenting work on behalf of many, many people. I've had many collaborators over the years that have made everything possible. In fact, they're not even all listed on this slide. But this is just to say that a lot of people have done a lot of work to make everything you're about to see possible. And so why me? I mean, I'm a dad, at least I used to be. I mean, I still am, but now, I mean, I've really had sort of a shift in career towards focusing on NGLY1 and other rare diseases and therapeutics for them. So how did this happen? Well, this is all due to Bertrand. So this is Bertrand back when he was, I think, about 2 years old. And life was difficult for him back then. We didn't know exactly what was going on with him and why he was having seizures, a movement disorder, a developmental delay, and for some reason a strange lack of tears. And it would have continued like that for much longer had it not been for a team of scientists at Duke University that intervened and said, hey, let's figure this out. And so this team here came to us and said, we'd like to do trio sequencing on Bertrand. We want to figure out if we can use the power of the genome to unlock the origins of Bertrand's health troubles. And so what they did is they sequenced me, they sequenced my wife Christina, and they sequenced Bertrand. They were looking for mutations that were driving his disease. And what they found was that he had in fact inherited 2 loss-of-function mutations in a gene called NGLY1. From me, he got a premature stop. From Christina, he got a frame shift that essentially wiped out the function of this gene. And then they said some interesting stuff. They said that we think this is the cause of Bertrand's disease. But if it is, and we're pretty sure it is, it looks like this is in fact the first case we've ever seen of this disease. There's no mention of this anywhere in the medical literature. So he's the only patient we know of for right now. And what I sometimes tell people trying to convey what this moment is like is that, I think every parent wants to believe that their child is unique. But there's some degrees of uniqueness that are far beyond what you really actually want. This was certainly one of them at the time. And so about 6 years ago, this is actually us getting the news at Duke University about 6 years ago. And I look back on this picture and I think, wow, how far have we come as a community from then to now? The other thing they told us, which I immediately disbelieved, that this was not actionable. That because there was just one patient, there was really nothing you could do for Bertrand. And I always knew that wasn't true. And I think you'll see now why, but I never believed that was true. And I think what happens in the absence of the not actionable, if you're being told there's nothing you can do, science itself steps in and becomes the action. So science, I think, becomes the kind of medicine that you take when there is no medicine to take. And this is something that I think is becoming a broader theme across the entire rare disease community. So, a few weeks after that diagnosis, we were out here in San Diego. We were meeting with Hud Freeze because that team at Duke had said, we're not glycobiologists. We don't really understand all the nuances of what's happening here. But there's a guy out in San Diego who might be able to help you. And his name is Dr. Freeze. So why don't you go talk to him and see if there's something you can do towards driving understanding and towards treatment and ultimately cure of this disease. And so what Hud really did for us when we met with him was he took us to the peak of our diagnostic journey. He said, this is how far you have come. in terms of getting an answer for Bertrand. And ultimately, this is how far you're going to have to go if what you want to do is achieve understanding, treatment, and cure. And so I was sitting in his lab several years ago and thinking, oh my gosh, that's a long way to go. But I think what all of us understand as rare disease parents and rare disease researchers is that you don't think about how far you have to go, you just think about what is the next step. And that's what we've all been doing for the past several years. So what is NGLY1? Well, scientifically speaking, NGLY1 is a gene that encodes an enzyme called N-glycanase, and N-glycanase is responsible for deglycosylating misfolded glycoproteins after they have been retrotranslocated from the endoplasmic reticulum into the cytoplasm. Obviously what that means is if you take this enzyme away, that you might expect to see an accumulation of misfolded glycoproteins. And this has sort of been one of the major operating theories behind the mechanism of harm in NGLY1 deficiency for really the past several years. But there's been a recent shift in perspective showing that there may be additional mechanisms at play, and that actually NGLY1 may be responsible for deglycosylating inert proteins and making them active, like NRF1. And actually, we have people like Gary Ruvkin to thank for this discovery. I mean, he wasn't necessarily looking for NGLY1 specifically. He was interrogating the proteasome and saying, well, what's interacting with the proteasome? And suddenly, in this genetic screen, up pops NGLY1 as an interactor. So, our understanding of the disease has evolved considerably in just the past several years. So, in terms of understanding, what kinds of things have we done? Well, we partnered with Hud Freeze almost immediately. I mean, within weeks, we were talking to Hud saying, how do we get a better understanding of this disease? Because at the time, there was a few obscure papers from Tadashi in Japan saying, this is sort of what we understand about the enzyme, but we didn't understand a lot about this disease at all. So Hud really kicked off a lot of those efforts. And then we got very lucky that shortly after that, that the NIH was very interested in setting up this natural history study for disorders of glycosylation. And Bill Gall and people like Lynn Wolf said, you know what, it would be really interesting to study NGLY1 as part of this. And so I know many of the families in this room have actually been through this protocol and understand what that's like. For those of you who haven't been through this protocol, just to give you a sense of what it is like to participate, and by the way, which I would encourage everyone to do, I've got a picture here for every poke, prod, procedure, or appointment that occurs during the course of this week-long admission at the NIH Clinical Center. It is an extremely intense experience. I think everybody leaves a little bit kind of broken and shattered by the end of it. And I say that, still inviting you to come because I think it's critical that everybody does this because it is really driving understanding for this entire family of diseases forward. And it's been nice for me because having been stationed in DC for much of my time, I always try to meet with families whenever I can when they go through. So it's been fun to see you all when you're there. And the other thing that this has generated is a tremendous amount of understanding about the disease, particularly in the form of things like biomarkers. So there's been a urine oligosaccharide discovered in the course of this protocol that serves as sort of a unique identifier for NGLY1 deficiency. And in fact, this is so unique it's sufficient to serve for a diagnosis. So there are patients being found through their urine instead of through just their genes for NGLY1 deficiency, all thanks to the work of the families and the scientists that have participated in this protocol. If you want to know more about this, there's actually a paper on this that describes much of what's been discovered through the course of this protocol. And in fact, for all the families that have been there and donated their literal blood, sweat, and tears, the cell lines are now available for public research through the Coriell Institute. So this is a major public scientific resource that's available to anybody who's really interested in studying NGLY1 deficiency. And so how do you go from understanding to treatment? Well, this is where things get trickier. But believe it or not, working with Hud Freeze, within just a few weeks of discovering this disease, we were actually taking credible, at least scientifically credible attacks on potentially treating the disease. And one of the first things we tried together was read-through therapeutics. So if you're not familiar with them, read-through therapeutics target a particular kind of genetic defect called a premature stop codon, which many of the CDGs and many of the patients with NGLY1 actually have as a driver for the disease. And the idea here is that when you see the premature stop appearing somewhere in the gene, you want to introduce a small molecule that will cause sort of a ribosomal skip over that premature stop and lead to full-length translation of the protein. So that the idea is to basically take the busted copy of the gene, read through this little stop sign that's been introduced by mutation, and try to get the full protein. And there's a class of compounds out there that are supposed to be able to do this, at least in theory. And so we tried them all out. I think there's about 4 or 5 of them, including things like gentamicin and G418 and atalurin. And unfortunately, none of them worked. But it was exciting, I think, at the time to even be testing compounds on cells so quickly after the discovery of the disorder. And it got me thinking, well, what else can we do? How far can we go with this disease in terms of the science? And so the next step was to really just try all the drugs. And this is, I think, a major thing that really every disease needs to do at some point is comprehensive drug screening. So to do this, you start off with your patient. You ultimately find the cause of the disease. You use that cause to generate some kind of model, usually a cellular model, though it can be an animal model too. And then you start combining this model with experimentation, something called an assay or some sort of experimental step that will give you a measure of what's happening with the disease. And through this, you can say, okay, we have a phenotype that tells us whether or not this disease is doing well or whether or not these cells are doing well or poorly with respect to this particular disease. Once you have that, you can start combining those models with different compounds in the hopes that one of them will actually reverse it. So the idea being that if you try enough drugs, and if you try essentially, say, the entire FDA-approved library, you might find one that actually works for your disease. And if you do, the hope is that you can take that back to the patients and actually use that as the starting point for a therapy for them. Now, it's not always the case that a drug that works in cells works in people. Sometimes there's a little translational process in the middle that you go through, and that's okay. And that's just part of the science that comes after that. And one of the hard parts for this, I would say, is actually figuring out the right assay to use to really interrogate that model system or the cells that you're using. And in this case, we have a few options for NGLY1. So there's an assay that directly measures the activity of NGLY1 itself, and so we're looking at that as the potential basis for a screen. But what I'm really excited about is this three-way collaboration between NGLY1.org, this foundation, and NCATS at NIH, and Retrophin, a pharmaceutical company, that have joined together to actually try to do these comprehensive drug screens, and in fact, novel drug screens, to look for the compounds that will actually make a difference for NGLY1. And what's amazing to me is just how fast a lot of this has happened, because I think we were just beginning to talk to Retrophin about 2 years ago at a meeting similar to this, and here we are with this actual collaboration set up today, looking for these actual therapeutics now. So there's other things you can consider in the course of treating a disease like NGLY1 or many of the other CDGs. You may want to consider enzyme replacement. And the idea here is, well, let's just synthesize NGLY1 and use NGLY1 itself as a drug. It turns out if you Google NGLY1, you can just buy synthetic human NGLY1 on the internet. And it's available for the low price of $402 for 10 micrograms. So it's not the cheapest stuff, but even so, it wouldn't be a terribly good therapeutic. If you were to just inject raw NGLY1 into a human being, it would be transported into the lysosome and degraded there. So it's not by itself, NGLY1 is not a drug, even though that's exactly what these children are missing. So what you really need to do here is some sort of medicinal chemistry on top of the enzyme, and in this case what you want to do is you want to attach polyethylene glycol anchors to the enzyme itself, and then on top of these attach something like TAT peptides taken from HIV, which make it cell penetrant, and so now you might have an enzyme that's converted into a drug from doing something like this. There's some other strategies you can use too when it comes to delivering an enzyme as a drug, but this is one of the ones that I worked on back at the University of Utah with some of my collaborators there. But what I'll say about that is a very costly, very lengthy endeavor. Enzyme replacement is not a cheap route to go when it comes to therapeutics. Something else you might consider is combing the entire literature to see if there's something out there that might help. And we've actually partnered with Andrew Sue and his lab at Scripps to do something like this through a project they set up called Mark to Cure. So what Mark to Cure enables is anybody that wants to volunteer their time the ability to go start marking up the literature surrounding things like NGLY1 and disorders of glycosylation in case there's something out there that we're not able to see. And what it allows you to do is construct knowledge graphs like this one right here that can connect concepts like NGLY1, perhaps phenotypically or symptomatically, if you will, through other diseases to other potential therapeutic mechanisms. And one of the first things to pop out of one of the original knowledge graphs produced by this was actually ACTH. And that sort of stood out to me right away because we'd already tried ACTH in Bertrand, so we knew what ACTH could do in the context of NGLY1 deficiency patients. And what it did for Bertrand was over the course of about a couple months is it completely normalized his EEG. I mean, he went from a child that seized virtually constantly to having no seizures, to when his neurologist saw the EEG, she said, I would think he's completely normal at this point. And that was kind of amazing because, and this is all pre-diagnosis, by the way, that really allowed Bertrand to be a person for the first time. I remember him smiling for the first time having gone through this treatment. So So the downside to ACTH, of course, is that you can't stay on it forever. It tripled his weight pretty much, and it more or less wiped out his immune system. So he ended up in the hospital for about 2 weeks after that. So that was not that was a very, it's unfortunately sort of a short-term therapy that we couldn't stay on long-term. But it has us wondering, well, what else is out there? And we're actively combing the literature now to see if there's anything else out there that might be amenable as a treatment for NGLY1. Something else that we've talked about yesterday, I thought Ava did a wonderful talk yesterday talking about potential dietary or supplementary strategies for these disorders is metabolic diets. And the core logic here that I often revisit when I talk to families or with a disease like this is if you're missing an enzyme that turns A's into B's and this suddenly goes away, then what's really happening here, what could be happening is that perhaps you have an accumulation of A's, a toxic accumulation of A's, or potentially a deficiency in B's. And so maybe the strategy is, can we modify the diet of these patients such that you end up giving B's or restricting A's or precursors to A's? And there's examples of diseases where this works quite well. So for PKU, if you restrict phenylalanine, it's a very effective treatment for the disease. There's even some CDGs, and in fact, in addition to the ones that Ava discussed yesterday, where this becomes an effective strategy as well. So for MPI CDG, you actually want to give mannose. So what about NGLY1? It's an enzyme. It has A's and B's, and actually thanks to work by folks like Tadashi, we have a really good understanding of what those A's and B's might really be. And so, using this sort of evolved portrait of the structure of the aggregates, I actually just took a guess that perhaps if there is a deficiency in something in NGLY1, it's actually represented here by this little blue square. And that little blue square happens to be N-acetylglucosamine. And so I thought, well, maybe this is worth trying in Bertrand. And so actually what I did is just to make sure it was safe, was actually ordered a bottle of it myself off of Amazon.com and just took the whole thing. And that for me, and when I didn't die, I just considered that FDA phase 1 safety testing. Still, there's sort of an ethical dilemma as to whether or not you give this to Bertrand, because it could have subtle interactions with his biology that we don't fully understand. And so we actually wrestled back and forth. Do we give this to Bertrand? Don't we give it to Bertrand? And he kind of sort of answered the question for us by himself, because he ended up hospitalized pretty severely with pneumonia at one point. And I remember sitting there thinking, if this bottle sitting on our shelf actually could have done something for him and he had died during that hospitalization, there's just no way we could forgive ourselves. So we sort of huddled as a family and we said, okay, once Bertrand's in the clear, once he's out of the hospital, once he's stable and able to withstand experimental treatment like this, we're going to give it to him and we're going to see what happens. So we did that, and about 3 days after he went on this compound, I walked into his room one morning and he was crying, and the amazing thing was he was actually crying tears. So in a disorder where these children have a very difficult time producing tears at all, there were tears really streaming down his cheek, and I was like, wow, well that is definitely different. I did not expect to see that. I didn't know what to expect to see with something like this, but tears are what we saw. And so that one little tear sort of started I think ultimately became an ocean of science for the disorder as a whole. And in fact, this is a true story. So we got on the phone with Hud Freeze and we said, Bertrand's crying, what do we do? And he said, well, what I want you to do is save those tears, pack them on dry ice, and ship them to me. So we literally packed his tears on dry ice and shipped them to Hud, because that's what you do when you're a family like ours. And you're going to hear a little bit more about this in a second, but we really followed up on the line of science that's opened by really studying it closely in flies. So you're going to hear more about this in just a moment from Clement Chow. And in fact, we even recently published a paper that gives us a better understanding of some of the mechanisms for why this might be a real therapeutic for NGLY1 deficiency. The question, of course, is can we do better? And thanks to folks like Tadashi, we always knew that we could. So Tadashi actually had some unpublished research, which he was very kind to share with the community to say, I think there's ways to do even better for these children. And that research is actually now published, and what he pointed to was a potential molecular target that could serve as the basis for therapeutics for NGLY1 deficiency. And the way this works is he was working with the mice, and he found that if you took NGLY1 out of the mice, they actually became embryonic lethal. They didn't survive. However, if you took NGLY1 and another gene called NGase out of the mice, suddenly they went back to surviving. So this is a very strong signal that if you could inhibit NGase, it could be therapeutic for these patients. And so I actually ended up applying for a small grant back at the University of Utah with some collaborators at the College of Pharmacy there and said, let's see if we can find these things. And the first thing we did is we verified that this was a real target. So we built a planarian worm model of NGLY1 deficiency. And the cool thing about planarian worms is that if you cut a planarian worm in half, what you get is 2 planaria. So they're a really good model of development if you're studying something like that, and this is certainly a key issue for NGLY1. So then we gave them NGLY1 deficiency by knocking down their NGLY1 with RNAi. And at this point, do you know what you get if you give NGLY1 deficiency to a planarian worm and you cut it in half? What you get is a worm that's been cut in half. So it turns out they lose this regenerative capacity when they get NGLY1 deficiency. But if you give them NGLY1 deficiency and you take out NGAs at the same time, and then you cut them in half, well, they regain this regenerative capacity. So this was an indicator that what Tadashi was seeing in the mice was translating into the worms, and at that point I thought would likely translate to the humans as well. So then the question is, well, how do you find an NGAs inhibitor? How do you find something that you can actually give to patients? And so, my original training is actually in computer science. I was a professor of computer science for a number of years, and I said, well, let's do computational simulations. So I partnered with folks at the Huntsman Cancer Institute. And what we did is we built a model of NGAs, and we did this by starting with a bacterial version of the protein. We did homology modeling to tweak it into what we thought the human version of the protein would look like. And then we did these simulations to see if there was anything out there, any small molecule, that was inverse in shape and charge to the catalytic domain so that it might serve as an inhibitor for NGAs. This is what we did. So we tested, I think, 200,000 different compounds computationally. 70 of those looked like they would actually have some effect as a potential inhibitor for NGAs. And 14 of them were already FDA approved. So at this point, we took all 14 of these into the lab and we used mass spectrometry to check to see if any of these really were true NGAs inhibitors. And one of them actually was. And that one is Prebys. So Prebys is a weak inhibitor of NGAs. And it turns out, in fact, that if we had published this result too, that all the proton pump inhibitors, of which Prebys is a member, also serve as NGAs inhibitors. So we have on the market therapeutics that potentially serve as NGAs inhibitors today. So the question I always get asked by NGLY1 parents is, should our kids take it? And my reply to that is, well, sure, why not? I mean, it's Prebys. So there's, of course, a word of caution to add to this, which is that you can't just take one pill to get the full effect. You probably need to take about 1,000 pills to get the full effect. And that's because at the concentrations where you achieve full inhibition of NGAs, you're at very, very high levels of Prebys, more than you would ever possibly take. And at this point, you would just wipe out your stomach acid. That said, you can get some levels of inhibition on Prebys at non-toxic quantities. And this could potentially be therapeutic for our children. So this is the actual IC50 curves that we actually see in practice. And so what we're doing now with folks back at Utah is some medicinal chemistry where we're taking the core backbone from proton pump inhibitors, this class of molecules that inhibits NGAs, and we're generating novel variants to see if any of these might actually be more potent NGAs inhibitors. And in fact, the latest molecule developed at Utah is now, I think, 60 times more potent than Prebys. So still not quite there. You'd still be taking an awful lot of it, but it could serve as the basis for a new therapeutic for NGLY1 deficiency. What about going beyond NGAs inhibitors? Well, it's again where folks like Clement Cho step back into the picture because he's got techniques as a geneticist so he can check for other interactors with NGLY1 that might ultimately serve as other targets for this disease. And so, for example, I think he'll talk a bit more about this, he can do something like a variation screen where you can rank the survival of different strains of fly that have been given NGLY1 deficiency to check to see which of these actually have modifiers in them that are impacting that survival and which might ultimately serve as the basis for drug development. So you could pull out genes from or variants from the high-performing flies versus the low-performing flies and say, well, maybe you want less of this and maybe you want more of this. That's the kind of stuff that could come out of the work that Clement is ultimately trying to do. So what's next? Well, for me personally and for NGLY1 as a whole, it's been interesting. So I had sort of a 2-year journey through the White House, and that was interesting. And I've also ended up as a faculty member at the Harvard Medical School and now as the director of the Hugh Kaul Precision Medicine Institute at UAB. And I'm using these positions as a way to really focus research directly on NGLY1 and many other rare diseases now. So the thing I'm most excited about at UAB is actually read-through therapeutics again, the very first thing we actually tried. Turns out UAB is a center of excellence when it comes to read-through therapeutics and has done large screens of molecules looking for read-through activity, and I'm going to target all that back at NGLY1 now because they've been mostly looking at these molecules for CF, but there's no reason we can't use them for NGLY1. NGLY1.org, which is a foundation that I've been involved with since its inception, is very actively engaged in supporting researchers. And I'll say right now, I'm very grateful for all the work that they've been able to do. And I can tell you that as an organization, I've never seen more science done per dollar donated than an organization like NGLY1. And on that note, since we actually have a number of the NGLY1 scientists here, if I could get you all to do a quick scientific huddle tomorrow morning at 8:30 a.m., I'd love to do that. If anybody else wants to come, you're certainly welcome, but with a huge warning that this is going to be a high throughput technical discussion that's not necessarily meant for families. That said, if you want to come, you're welcome. If you want details on this, please talk to me or to Carrie about where to go at 8:30 tomorrow, just so we can sort of sync up and figure out where to go next. So a few special thank yous before I depart, which is, of course, I have to thank my parents for sponsoring a lot of the initial research into NGLY1 deficiency and really kickstarting this entire effort. So without them, really none of this would have been possible. And of course, to my family for putting up with all my crap for the past 5 years. Because I've been on something of a personal and professional odyssey as I moved from computer science into medicine and, really now sort of rare disease therapeutics in general. So with that, I'm sure I'm out of time by now, but I really just want to thank you all for being here and remind you that I think that the core message at the end of the day is that I'm sure we've all been told, like we were many years ago, that this diagnosis that you've received for your child is not actionable, except that was never true, because in the event of the not actionable, it has always been possible and always will be possible to do science. So thank you.
Questions? Is there any phenotype associated with the heterozygote?
That's a good question. I'm guessing some of the other speakers may be addressing this. So the short answer is we haven't found one yet. But there's some evidence that it might confer some resistance to certain classes of viruses. I think enteroviruses you might have some advantage there, but there hasn't been much of one that we've seen so far.
On that very important note, part of the cell lines that are now available at Cornell are carrier cell lines. So every single parent and the patients have lymphoblast cultures and fibroblast cultures. And so I think some characterization of carrier parents could be very illustrative to researchers, which is part of why the parents' tissues were also sent.
Thank you. Anything else? Okay.
All right. Thank you.
Thank you again.